Well, thank you, Riching, very much for the invitation to come here. Particularly, I like speaking to groups of people that I may not necessarily have spoken to before, and hopefully I can keep Bruno awake. If not, I will try not to take it too personally. But since I am somewhat new to this particular group, I thought I would maybe just give a two or three quick introduction about where I work and what we do. But I will try to switch very quickly from there and talk about some of the tools that we're using for visualizing and processing and even some analysis of large 3D EM data sets here at Janelia. So for those of you that don't know, Janelia Research Campus is the flagship research campus for the Howard Hughes Medical Institute. We are just up the road from Washington, D.C., not too far from where Wuching is. Let me get my laser pointer. Just up the road from Dulles International Airport. And in particular, I work in, and this is a nice aerial picture of our campus. So what I work on at Janelia is in a center called the Advanced Imaging Center. And so what that is it was started about 2014, and it was started with the goal for sharing some of the imaging technologies that were developed at Janelia to the wider world. And so what that means practically is that people can access the AIC through a proposal, and if your proposal is accepted, visitors usually come and visit Janelia for somewhere between 2 or 3 weeks at a time, and we cover the cost of your experiments, including most of the basic reagents. You get free scope time, and you get free access to one of the really smart people that work with me at the AIC who are dedicated to your project full-time while you were there. And we'll even pay for your lodging. This is a picture of the hotel that we have at Janelia. We have a number of different microscopes in the center. I'm certainly not going to go through each and every one of these, but a lot of what I'm going to talk about really sprang forth from the fact that the AIC has a version of the enhanced focused ion beam SEM that was developed at Janelia, primarily in Harold Hess's group, that we make available to people who want to use it. And through that experience, we've really had a kind of a crash course in understanding what are some of the really best tools that we can use to deal with the absolute mountains of data that can come off of this particular scope. All of these scopes generate enormous amounts of data, but I'll focus specifically on some of the tools that we use in this context. Like I said, I don't necessarily want to go too in-depth into the program of the AIC. You guys are more than welcome to check us out on the website or just send us an email. We love talking to people. If you want to know more just in a general sense, if you want to talk about it in the context of a specific project, either way is fine. So, much like a lot of different biological imaging techniques, EM is no different in the sense that there's a realization and a prioritization of trying to gain 3D information. As we all know, biology doesn't exist in 2D, as informative as 2D images can be. And so, I have just three, I think, common examples of how people are using EM to gather 3D information. There's, of course, serial section, TEM, where we very laboriously section a sample and image each of those sections in sequence in the TEM in an effort to build up a 3D block of information. There's, of course, the tomographic methods, which are a little bit different, but use principles of tomography to get a 3D picture of what we're looking at. And then, of course, there's the serial block face sectioning SEM type of techniques where the sample is put whole inside of the microscope and is sectioned serially and imaged as we go. And all of these techniques are certainly becoming more and more popular in many contexts. And I think if we kind of think about kind of their pluses and minuses in this kind of diagram, I think it kind of makes sense a little bit to me. So, the tomographic methods, I think, tend to give you really exquisite detail. We can get down into 1 to even less nanometer resolution, certainly, but the volume that you're going to be able to image is likely to be more limited, again, because of the limitations of tomography. The sectioning sort of base techniques like serial section or block face tend to kind of exist on another side of this plot, where the resolution, at least in the cutting direction, can typically be limited just by the physical properties of the sample, but that is in a sense made up for by the fact that we can image larger and larger volumes. And so in many ways, these two types of technologies are very complementary to each other. Now, a lot of the work at Genelia over at, say, the past 10 or 12 years has been looking into this fourth technique called FibSim. And I'll show a slide here in a minute if people are unfamiliar. But that, I think you can kind of say, exists sort of as in a middle ground. On the one hand, we can typically get better resolution, particularly in that axial direction, that Z direction. But historically, have been fairly limited in the amount of volume that you can get reliably, and a lot of the work at Genelia has been trying to push the capabilities of FibSim to image longer at a time in order for us to image higher amounts of volume, and that's really what it comes down to. So the picture on the left here is the AIC's FibSim system that we have. It is I guess you could call it sort of a hybrid commercial and bespoke system. It works off of a commercial SEM system, but we've engineered very specifically some things into the system, including part of the fib that increases the stability over what some other commercial systems can achieve. But the basic idea is relatively simple, and it's pretty analogous to a serial block face in the sense, but instead of using some kind of physical sectioning, what the system will do is use a focused beam of ions, usually gallium, to ablate the surface of a resin-embedded sample, and then we will come in with an SEM and scan our way through, create an image, and repeat over and over and over again. So a lot of the work being done at Genelia has been, how do we make this process repeatable for thousands, tens of thousands, maybe hundreds of thousands of images? Can we create a system that is stable against disruptions? Can we create a system that is able to ablate a very predictable amount of your sample at every slice, so that our resolution in the Z direction isn't changing dramatically from one slice to another? And so that was a big effort, again, primarily through Harold Hess's lab here at Genelia, and he had an excellent scientist named Shansu, who is now at Yale. But one of the big ideas that they had was to actually create a lot of feedback loops between what the fib beam was doing and what the sample was experiencing, and being able to control that in a very, very tight way. And along with that, there's been a lot of engineering going into creating a system that would be responsive to things that we can't predict very easily. If the temperature, if there's a temperature excursion in the room, we want to know that. Or if there's a rapid drop in image quality as we're taking images, we'd want to know that. And so all of these things are sort of built in to allow the system to stop gently if there is a mishap, giving us a chance to then hopefully fix the problem and then start the system back up, hopefully with very little or no data loss. And so that, in the end, is what really allowed them to be able to image a sample for months and even years at a time, if need be, because that, unfortunately, is the biggest drawback, I think, with the FibSim imaging techniques, is that they're relatively slow. So we at the AIC, we're very agnostic in terms of what kind of projects that we like to have. If it's interesting, we're more than willing to talk about it. This is just an example I wanted to show of a malaria oocyst that's actually developing inside the mosquito gut. And we were able to get wonderful data, but even more importantly, we were able to visualize and even segment some of the structures that were inside of this parasite. And I wanted to kind of now transition into really the focus of my talk, which is how do we go from taking raw data off of the microscope, thousands, tens of thousands of images. How do we deal with that? How do we go through and process these? How do we go through and visualize them? And how do we try to extract some quantitative information? So that'll kind of form the three parts, I think, of my talk here for hopefully not much more than 35 minutes here. My goal here is to really kind of give everyone a survey of some available tools. I would love to go really in depth into any one of these topics. I think each one of these could probably be its own talk, but I really want to kind of give people who may not be aware of what is out there, what is available to us. And so I'm going to talk a little bit on the processing side. I'll talk a little bit about how we register our data, how we can try to process the data to reduce the influence of noise in the images. And I'm going to talk about how do we view these really extraordinarily large data sets. A typical desktop computer is not going to be able to even load this amount of data. So how in the world are we going to go through and look at this? And then finally, I'm going to talk a little bit about some machine learning image segmentation tools that we commonly use here. I'm going to give two sort of contrasting examples here that I think might be useful if you're thinking about getting into that sort of thing. But really, the things that I really want to make sure that I do is emphasize that all the tools that I'm going to talk about here are open source. In other words, they're freely available to anyone with an internet connection. And for the most part, all of these tools are capable of dealing with what we lovingly call big data, and that has lots of different definitions. But for us, big data, from a quantitative standpoint, could mean hundreds to thousands of gigabytes. But really, to me, it means data that is too big for you to load into the memory of the computer that you have. And most of these tools have ways to get around that problem. So let me talk briefly a bit about one of the fundamental things that almost everybody has to do in 3D EM microscopy. And that's this problem of image registration. Now, of course, this doesn't necessarily apply to tomographic techniques, but any of the sort of sectioning techniques, this is going to be something that you have to do. And so if I were to take all of the slices that we took, say, in a FibSim, but this could easily apply to serial section or blockface, if we were just to naively put them on top of each other and then look at it from the side, so the Z direction here is the direction that we were sectioning our sample, very likely you're going to see something like this, which is, it's just not going to be informative in the slightest. And what we need to be able to do is from one image to the next, we may need to shift it, we may need to rotate it, we may even need to scale it slightly so that if we were to look at it from the side, that we would be able to understand what are the structures there in our images. And so it is a difficult problem to address. In the past, many people have thought about, are there ways that we can introduce some kind of fiducial marker into our sample that would act as sort of an unbiased way for us to register the images from one slice to the next? Not always a very practical approach in every instance. And so what we're really forced to do is say, okay, can we look at one image and look at the next image, and can we understand how to align the two of them together? And that really requires us to find a robust and reproducible way to find some kind of feature in the image that exists between one slice to the next that we can use as a way to align two images. And, our brains are great at doing this. We can look and see, oh, this is clearly the same image. It's just been shifted and rotated slightly. But how do we get a computer to do this? Well, there's lots of different ways that we can do this, but I'm going to talk about the one I think is probably most commonly used. Doesn't necessarily mean that it doesn't have its weaknesses, but I think it's the one that if you were going to look more deeply into this topic, this is probably one of the first things that you would see. And so this is called the SIFT algorithm. It stands for Spatially Invariant Feature Transform It was proposed by a guy named David Lowe, a number of years ago now. It's a very powerful technique in that it finds features in an image that are distinct, that we can use then to match similar features in the next image. And the way it works, without getting too deep into it, is that we take an image, for example, and we do a couple of different things to it. One thing we do is we will start progressively blurring that image. And then we can also progressively rescale that image, so we can make it smaller and smaller. And by comparing the differences between these blurred images, we can use that to as a way to find areas in your image that show very clear edges or discontinuities, which are good indicators that there's some kind of interesting object there. And if we can kind of compare what we get at different scales, we can start identifying features in an image that aren't going to go away too much if we have to maybe rescale or, remagnify your image or rotate them around. And also at each one of these features, we can also sort of calculate what is the gradient around that feature in a way to kind of infer an orientation of that feature, which is a little bit nebulous to think about, but you can sort of think about it, in the sense that if we found a feature in our image, say our little melonhead cat here, clearly his eyes and maybe his nose stand out as very clear features in the image, and perhaps just the way that the image is oriented, we can say that these features have some directionality associated with them. And if we were to then do the same thing in our rotated image here, that gives us a very robust way for us to match one image to the next. And what's great about this is that it actually comes standard in one of the most common image analysis and processing packages out there, which is Fiji. If anybody has done much exploration in the plugins folder, if you go down to the registration, there is a lovely implementation of the SIFT algorithm in Fiji. So if this is an issue that you're facing with some of your data, I think this might be the first stop to see can you try the SIFT algorithm to see if you can get the sections of your images better aligned with respect to each other. Unfortunately, this particular approach is not going to always work well with really big data. And so one of my very close colleagues here at Genelia has actually put together, I think, a really nice implementation of the same algorithm, but really geared towards addressing the problems that come with really big data. And so this is included as a Python script. If that's something that is a little bit nerve-wracking for you, it's rest assured it is very well documented how to install it. You really don't need an extraordinarily powerful computer to do this. And he includes a lot of sort of examples and how to set this up. And so, even though this was written for the Genelia FibSim data in mind, it is very applicable to other kinds of 3D IM and frankly any kind of 3D imaging technique in principle. If you're faced with a relatively large data problem that you need to do some image alignment, I think this is a good option. Certainly not the only one, but one that I can personally attest to seems to work very robustly. Okay, so I want to touch briefly here. I'll try to go through this too quickly so we have time to talk about some of the other things. But as we all know, and this isn't certainly unique to electron microscopy, but I'm going to give an example here. We often have to make compromises when we're imaging, either because of less than ideal sample preparation. Maybe when we're doing 3D imaging, we don't necessarily want to burn our samples in order to maximize the quality of the image. And so it is a fact of life that many of us are going to be faced with images with relatively limited quality or limited signal to noise. And in the age of machine learning, I think this is becoming a problem that can be addressed fairly robustly using quantitative methods. Now, I know that probably most of us are at least somewhat familiar with ways to make the noise in your image appear less. There's three just common examples. they're not always very ideal, right? If you do a Gaussian blur on an image, sure, we can quiet down the noise in your image, but very often you're also going to blur the structures that you're interested in seeing. A median filter maybe won't do that as much, but because of the sort of the nonlinear nature of how a median filter works, you can often kind of get this modeled effect when we look at EM. And there's even some more sophisticated methods that have their pluses and minuses too, but uh, it's not unusual for us to struggle in this realm. And so what I wanted to point out was just one particular package that I think is very useful. It's been around for quite a long time, and it's called noise to void. And what's great about noise to void is that although it is a machine learning technique or machine learning algorithm, it is the way that it is set up is that your data itself trains the model, and so you don't necessarily have to have what you would call ground truth. That is a necessary component for a lot of machine learning these days. And so what it does is it actually uses the noise statistics that are already there in your images to try to infer what the underlying structure in your image is. And here, I think this was a nice example where it looks like they were doing some cryo-ET. Really be able to pull out some features in your image surprisingly well. And so, this is just an example that I did very, very briefly. If I take this same data that I showed earlier and apply the noise to void to it, my sense is that it does an excellent job. It does an excellent job of both reducing the noise content while trying to preserve the underlying structure. And these yellow lines, you were probably wondering what they were for. Well, I actually wanted to see, quantitatively, how do these different denoising techniques work? And I think I won't belabor this too much, but I think the noise to void model does, by and large, a very excellent model. Like any machine learning technique, the results are going to depend heavily on how much training you're willing to do. But I think that's a general truism. This is also, of course, a freely available piece of software. You can do it in Fiji as well, so you don't necessarily have to go into the Python world and deal with package managers and anything like that. You can do this from the comfort of Fiji. Okay, so I want to go ahead and switch topics here briefly before I go to the last bit, and that's the issue of visualization, which may seem a little bit fundamental, but I want to give an example to give that might give an idea of the scale of the problem. So this was a piece of FibSim data that we took at Janelia. This was a part of the Drosophila optic lobe. This piece of data took 100 days to acquire, and it represents, I think it's tens and tens of terabytes. It's an extremely large piece of data, and this is far beyond the capacity of even a really pretty good computer for us to load all of this data into and simply scroll through the slices to see what we have in an image. And so what do we do in a situation like this where the data is so enormous that even a relatively good computer cannot possibly ingest it all at once? Well, as it happens, a lot of the commercial software developers out there, like Amaris, like Amira, they do have capabilities now to deal with this problem. But what I want to focus on is actually a freely available way to do this. And that's called Big Data Viewer. And this is led in part by some computational folks here at Janelia, as well as in Max Planck and University of Wisconsin. This comes freely available in Fiji. You download Fiji and it is instantly available to you. You don't even have to install it. And the way that all of these implementations work is through this concept that we lovingly call lazy loading. And it's through kind of two different ways that we manipulate the data to that allow us to be able to view it sort of arbitrarily without having to load the entire data set, 10 terabytes of data into your memory. So relies on a couple of different things. First, it's going to take a 3D data set and divide it up into little chunks. And that's exactly what they call them. They call them chunked data sets. So we're going to divide, if you can imagine your 3D image stack is just a big 3D matrix of numbers, we're going to divide those up into little clusters, say not more than maybe 128 pixels by 128 pixels by 128 pixels, for example. And so what that allows us to do now is if you do this in the right architecture is that you can tell the computer to only load the parts of the data that you're looking at. So if we are only looking at a particular corner of your data, it's only going to load the part of the data that you are looking at. Now, of course, you might ask, well, what if you want to look at the whole image at once? Well, the second thing that it's going to do is actually going to group these individual chunks of data into larger chunks of data, but we're going to downsample them. In other words, we're just going to bin the pixels. So now we still have the same number of pixels, they just have a worse resolution. And we'll do this maybe twice, maybe 4 times, maybe 8 times, and build what we call a resolution pyramid. And so what that means is that when we are looking at a relatively large image far away, we're zoomed out so we can see the whole thing, your eyes cannot necessarily perceive at the pixel level what the details are anyway, so we can get away with having a worse resolution image. If you zoom in, you're by necessity looking at a smaller part of the image anyway, and that's when the larger, the full resolution of the image then is loaded just in that particular part. And so this is a way for us to get around having to load enormous amounts of data into your computer. And so just so you guys can see what this looks like in reality, I just took a little screen capture of myself playing with some data last night, so you can see that this really doesn't indeed work surprisingly well. So this is a FibSim data set that we had taken some time ago, and I can very quickly sort of scroll through some of the slices, and people that come to visit us here at the IC do this all the time, and they say, oh, okay, well, maybe I want to look at this nucleus here. I love nuclear pore complexes, so we can zoom in. And you can see as we can zoom in, hopefully you guys can appreciate from the movie here that it's progressively loading the image in more and more detail now that we're trying to view it more close up. And we can look at it very easily from different orientations too. We can look at it from the orthogonal views, and we can even slice the data arbitrarily. So if we wanted to just look at it sort of at an odd angle here, we can do that. So that's what Big Data Viewer can do. I think it's a great software package, freely available, and very, very powerful for people to really just be able to explore their data, try to understand what exactly it is we're looking at. Okay. All right. So I will hopefully here in the next, let's see how much time I have, about 15 minutes. I will talk a little bit now about machine learning segmentation. And I know this is a big topic. This is something that's the subject of enormous amounts of effort in many, 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 many different fields. And I want to try to parse out exactly what it is we're trying to do with machine learning segmentation, particularly in the context of 3D EM. So there's even a team project here at Genelia called the CellMap team that's just dedicated specifically to developing tools and techniques and to gathering a repository of 3D EM data that has a machine learning based segmentation as part of that data. So if this is just a very small snippet of some FibSim data, I believe it was a HeLa cell. We want to be able to take these images and create organelles out of this, and to say then further on, can we say something fundamental about some of the images that we're looking at from a biological perspective. And this is certainly no mean feat. And had you talked to me about this maybe 10 years ago, I think I would have laughed in your face and said there's no way that this is possible. Our own brains, our human brains oftentimes don't often know at all times what we are looking at in an EM image. But, excuse me, the advent of machine learning has changed, a lot of what people think is possible in terms of extracting information from EM images. And then, it should come as no surprise, therefore, that there are just a ton of uh, machine learning segmentation software out there. And most of the commercial software developers also have their own implementation. Excuse me. Um, I think all of these have um, pluses and minuses to them. All of them have different. Excuse me. All of them have different um, use cases in mind. I think what I'm going to do is, rather than going through all of these, I'm going to focus on two that we use commonly here at Genelia. So I want to talk about two packages that I think are excellent examples, I think, of what are out there. And that's one of them is called Elastic, and another one, which I love the name, is called Empanada. Let me start with Elastic first. Um, Elastic has actually been around for a long time, more than 10 years. The thing that I think is really powerful about this particular piece of software is that it's really geared towards users who have really little to no machine learning experience. So for a lot of people who are interested in learning how to do machine learning segmentation, I think that this is a great place to start. It's freely available, obviously, as everything is that I've been talking about here. What's interesting is you can install it as its own piece of software, but it also has integration with Fiji. And so depending on what you might be wanting to do with it, you can choose either or very often we'll do both implementations of Elastic. But let me just talk a little bit about the software itself and why I think it's a useful thing to try, especially if you're first getting into machine learning segmentation. First of all, it can handle big data. It has a particular data format that it uses. Unfortunately, it's not quite the same format as the big data viewer, but you can actually convert your data from one to the other quite easily. One of the nice things about Elastic, I think, is it gives you a flexibility in what features that you want to tell the computer to look for when it is trying to find objects that you are saying are there, which I think is somewhat rare in a lot of machine learning softwares. So you can, by default, just say, I want you to try every possible means to use your machine learning capabilities, or if you want to try a few different features, you can do that. So I think that it's a nice thing. It's also a nice way to kind of understand really how machine learning works, because I think for a lot of people it is a bit of a black box. But at a fundamental, perspective, machine learning is just about recognizing patterns, and how we recognize these patterns is based on these features that the machine computes for every pixel in your image. So understanding how those features relate to a predictive ability, I think Elastic has a nice way to do that. So again, you can pick any and all features that Elastic uses, and then we just tell you have to give the computer examples of what is interesting and what is not interesting in your images. And I know this is not an EM image, but I think it's simple enough to understand. If we wanted to ask Elastic to tell me where are the cells and where's the background in this image, we really only have to give it a few representative examples of a cell, very quickly just paint what a cell is in blue and what the background is in yellow, and very easily it's going to be able to pick that out. A very powerful technique, and this is something that would be, frankly, a little tricky to implement using sort of traditional techniques like thresholding and things like that, because again, for example, you have different morphologies of different kinds of cells at different stages, and so this is something that machine learning is really quite excellent at doing. But like anything else, it has limitations. So I want to spend a few minutes talking about what's good about Elastic and where you might want to start looking at some other possibilities out there. So, like I said initially, it's an easy to use piece of software. very trivial to install. And what I really appreciate, which is not always the case in a lot of open source software, is that the documentation for Elastic is excellent. There's a lot of tutorials in there. They have a very intuitive way to understand how this software works. And incidentally, I won't have time to cover it, but Elastic also has a lot of other workflows in it. It's not just used for machine learning pixel classification. There's a lot of other functionality inside Elastic that may be useful for your projects. Like I said, it also has ImageJ integration. So if you have a big data set, there's a plugin you can use. You can load up your data, and it will convert it automatically to a format that works best for Elastic. But if you don't have an enormous piece of data, you can load TIFF files or any of the other proprietary EM files formats into it, no problem. So like I said, it supports large data. It uses that similar kind of lazy loading architecture that I talked about earlier, where it divides your data into smaller chunks, and then sort of scales it depending on how zoomed and zoomed out you wanna view your data. And what I really appreciate about it is that it takes a very sort of old school wizard-like interface. It just takes you through step 1, step 2, step 3, step 4. It's very hard to get confused as to what step you're supposed to be doing next with Elastic, which I really personally appreciate a lot, especially as someone who works in an imaging center where you have a lot of people coming in that this may be their first time using any kind of machine learning at all. But, I think after a while you may come to find that there are some limitations like any software has. For one, the blessing and the curse of Elastic is an older piece of software, which means it is very stable. It's it handles a lot of the usability issues that most software will encounter very well. But it also has an older machine learning architecture. So in other words, the technology is progressing so quickly that the underlying technology at this point is frankly starting to show its age. And so in part because of this, it can require quite a bit of training data for you to get really good results, especially if you're trying to pick out really subtle features in your images. You're going to have to tell Elastic, you're going to have to give it a lot of examples of what that object is going to look like. So that can require a lot of time. And because of the architecture that it uses, it's really going to work best if you go through and you just label everything that you think is interesting in your image at once. And that's not always an easy thing to do, because very often our understanding of what the images looks like sort of evolves over time. So rather than saying, okay, I just want the computer to find where all my mitochondria are or all my microtubules are, it will really work best if you kind of tell Elastic, these are all of the things that I'm interested in. And that can be difficult to do sometimes, especially if you're still kind of evolving your understanding of what the data is telling you. And lastly, it can be particularly slow, again, because of the older architecture that it uses. So with large data, it can take quite a long time for it to spit out the results that you're interested in just from a computational point of view. So if you're interested in really, really fast iterations and trying to iterate your way through a machine learning workflow with large data, Elastic is not always an easy way to do that, but extremely user-friendly. Now, I want to contrast Elastic with another piece of software that we've become pretty interested in recently, and this is called Empanada. It stands for EM Panoptic Any Dimension Annotation. It's only been around for, I think, about a year and a half, but from what I've seen so far, it is actually an incredibly powerful piece of software. It's not its own standalone software. It exists as a plugin for another package called Napari. And so for anybody that's not familiar, Napari is another open source image viewing software that was particularly supported by Chan Zuckerberg Initiative. But was originally kind of, I think, kind of meant to be a competitor to ImageJ. I don't think it's going to be that anytime soon, but it is a commonly used image viewing program out there that is freely available. But Empanada requires, I think, a bit more knowledge. So if machine learning is not something that you've done, this is probably not the first thing that I would go with unless you really have someone there to kind of help you interpret some of the results and help you guide you along the way. But it does have a very flexible sort of modular interface that really allows you to be more innovative in how you want to do things. But I think the big fundamental difference between Empanada and Elastic is that Empanada does what's called panoptic segmentation, which means it uses both semantic and instance segmentation. So what does that mean? Well, we'll do something simple here. Let's say we have an image that was taken on the street. Now, what a semantic segmentation is going to do is going to say, okay, the computer, tell me where all the cars are and color them blue, and that's what the computer did, and tell me where a building is, where's the road, and it's going to give each of those objects a certain color, a certain identity. What instance segmentation asks is, don't just tell me where all the cars are, but tell me where individual cars, or people for that matter, are. And what panoptic segmentation refers to is like a combination of the two, where it makes sense to think about things as individual objects, do that. Where it doesn't, don't do that. So maybe I'll give a more intuitive example from a biological point of view. So the semantic segmentation makes a lot of sense if, say, we're trying to find where all the ER is in an image. ER is sort of it's a distributed organelle. There's not really such a thing as or ER. It forms a tubulated network in the cells. On the other hand, mitochondria, it makes sense to think about those as discrete things. There are individual mitochondria distributed throughout the cells. And so what Empanada does is it allows you to do both kind of back and forth seamlessly. And that's powerful, something that Elastic does not offer. Okay, so when we're talking a little bit about the pros and cons of Empanada here, it uses a much more modern machine learning neural network architecture. So it is powerful and it is fast. Probably, I would say, 10 times faster than Elastic. The documentation, I think, is still quite good, and there are a lot of good tutorials online that you can go view that I think would do a great job to get started. It still can support large data, multi-hundred gigabyte, terabyte level data through Napari. Like I said before, it is very modular and flexible, so you're not necessarily stuck with a single workflow if for some reason that doesn't make sense to you, given what project. And like I said, it has this panoptic segmentation, which is conceptually a very powerful thing. But as you can probably guess, the cons are in many ways the mirror opposite of Elastic. It's more complex to install. It's going to require you to be at least somewhat familiar with Python package management and things like that. One of the things that I think really needs to be improved in this software is its error handling. If you click a wrong button or input a parameter incorrectly, without having a fairly good understanding of Python, it's going to be difficult to know what you did wrong. And that's, I think, one of the biggest hurdles I think this software is facing at the moment. But in a general level, this wouldn't necessarily be the software I would go to if you're not familiar either with machine learning or Napari. If you do want to use this, take a little bit of time and learn a little bit more about the underlying technologies that support this software. I would say would be a good idea before you jump right in. The workflow also is it's not this nice wizard step 1, step 2, step 3 interface, and so you really have to understand exactly which module is appropriate to do at which point in the process. And like any new software, development is ongoing. So you can expect changes that can be a little bit disruptive, as you would in any software. So actually, as it happens, I think that was my last slide. So I think we can go ahead and take questions whenever you're ready.